My name is Jennifer Cobble, and I'm the Executive Director of SMAA. We regret that we can't meet you in person this year, but we're very excited to welcome over 100 of you who are joining us virtually from all over the nation to celebrate your connection to Stanford School of Medicine and our broader Stanford alumni community. Before we begin, I'd like to remind you a few things about the technology of working on Zoom. First of all, I'd like to remind you that your microphones and your visual profiles have been set to listen-only mode. Only the speaker and the moderator will be on your shared video screen. Should you encounter technical difficulties, please try leaving the seminar and logging back in again. But if issues persist, you can reach out to us at medalumni@stanford.edu. That's medalumni, one word, at stanford.edu, and one of our staff members will try to help you. Finally, we will be answering questions at the end of the presentation, but you can submit your questions anytime during the presentation by using the Q&A feature at the bottom of the screen. So if you scroll down to the bottom of your screen, you see over in the right-hand side of the middle of the screen, a Q&A. You click that and then you will be able to type in your question. If you see a question similar to your own, you can like it by clicking the thumbs up icon, and that question will automatically move up in the priorities. Unfortunately, given our large audience, I regret we can't always ensure that all questions will be answered in the time that we have allotted, but we will try to group questions and get to as many as we can during our time frame. So now I am pleased to begin our program and introduce our Stanford Medicine Alumni Association Board of Governors President, a graduate of the class of 1983, and the Chief Medical Officer at Sutro Biopharma. Please welcome Dr. Arturo Molina.
Hello, everybody. Good afternoon. On behalf of the Stanford Medicine Alumni Association and the SMAA Board of Governors, I would like to welcome you to our annual half-century event. As Jennifer mentioned, because of the pandemic, we're doing this event virtually this year, so bear with us as we navigate through this together. Before I introduce our guest speaker, I would like to call out some of today's alumni participants. Representing our most senior alumni classes are Cy Kinsell, Dr. Kinsell, class of 1947, Dr. Richard Wilbur, class of 1947, and Dr. Thomas Leo, class of 1948. So welcome. Representing the guest who has logged in from the furthest location is a lot of you. So the benefit of doing this event virtually this year is that we have over 50 alumni logging in from out of state. I cannot list them all, but we'll say we are represented in such states as Pennsylvania, New Jersey, Maryland, Washington, D.C., Virginia, Illinois, Arizona, New Mexico, Hawaii, Texas, Wyoming, Georgia, and even Canada. So welcome all. And I would also like to welcome those who are new to the Half Century Club, having celebrated their 50th reunion last year. Again, there are too many to name, over 20 of you joining us today, so I will just welcome all of the MDs and nurses from the class of 1970. So welcome, and I hope you enjoy the program. I would now like to introduce Dr. Ewan Ashley, who is Associate Dean at the School of Medicine and Professor of Medicine, of Genetics, and of Biomedical Data Science. Born in Scotland, Dr. Ashley graduated with first-class honors in physiology and medicine from the University of Glasgow. He completed medical residency and a PhD in molecular physiology at the University of Oxford before moving to Stanford University, where he trained in cardiology and advanced heart failure, joining the faculty in 2006. His group is focused on the science of precision medicine. In 2010, he led the team that carried out the first clinical interpretation of a human genome. He now routinely applies genome sequencing to the diagnosis of patients at Stanford Hospital, where Dr. Ashley directs the clinical genome program in the Center for Inherited Cardiovascular Disease. Dr. Ashley was the first co-chair of the steering committee of the NIH Undiagnosed Diseases Network. He was a recipient of the National Innovation Award from the American Heart Association and the NIH Director's New Innovator Award. He is part of the winning team of the 75 million One Brave Idea competition and co-founder of 3 companies, Personalis, DeepCell, and SVXA. He was recognized by the Obama White House for his contributions to personalized medicine, and in 2018 was awarded the American Heart Association Medal of Honor for genomic and precision medicine. His book, The Genome Odyssey, is scheduled to be released in February in this month. So we're very honored to have Dr. Ashley. Dr. Ashley, welcome.
Well, thank you so much, Arturo, and thank you to Jennifer and the whole team at the Alumni Association. Welcome to this unusual version or something that is now perhaps more usual for us, which is a Zoom presentation. And I hope that you're sitting somewhere comfortably and perhaps you have some lunch, depending on your time zone, or something else, depending on if you're a little further on in the day. It's great to be here, and I'd love to talk to you over the next half hour about some of the work that we've been doing at Stanford relating to human genomes and their ability to help us prevent and predict disease. But first of all, before we get to that, I think I need to answer this question for you, which is, who is this guy and where did he come from? And perhaps, given the photo on the screen, we need to do better than that and answer the question, what is he wearing? And that, in order to answer those questions, let me take you not just to where I am now, which is what you've heard already in the introduction, but to where I began. And this is in fact Scotland, and I'm originally from the island of Britain and the country of Scotland. I wasn't actually born on the site of Lafroig Distillery. Some of you may be familiar with this as a single malt from the west coast of Scotland, the island of Islay. I'm actually from Glasgow, as Arturo mentioned, that one of the largest city in Scotland. But this is my family. We visited just a year or two ago, the island of Islay, and I certainly have a sideline in research on the health benefits of single malt Scotch. Not going to talk about that today. But I did want to tell you a little bit about how I ended up at Stanford just before we get going. This is me with my father, who was a physician in a local town in the west of Scotland. This is what counts for a swimming pool in the west of Scotland, where it rains many days out of the year. This is one of the sports, fishing. I look rather cold in this photo because in general it's pretty cold in Scotland. Let's see if we can keep the slides moving forward. I had aspirations like most boys do to superheroes when I was young. It's a picture of me in a Spider-Man outfit. I learned the Rubik's Cube when I was about 12 with my father and started programming computers around the same time. And I didn't imagine at that time that the skills I was working out trying to create computer games would one day be useful for thinking about human genomes and their effect on health. But my main inspiration for going into medicine was my father, who was a local GP, as I mentioned, shown here. He actually is called Angus, and that's my middle name. And my mom, shown here in our front hallway in our home, was a midwife. And so I grew up in a medical family. I used to go on house calls with my dad and my mom, actually, and we lived in between a fairly affluent area and a very poor area. In fact, my mom worked in the poorer area, and I would go on calls with her, and she'd see early teenagers on their second pregnancy. My dad worked in the slightly more affluent area, and I would go and see the contrast, the importance of social aspects of medicine. But that took me eventually, in thinking about the cutting edge of medicine, to Oxford, where I did a PhD. This is Christ Church College, now as well as much else, better known for having the site that you see in the Harry Potter movies, where all the kids line up and have their desserts and feasts. This is the main hall in Christ Church that it was actually based on. But I took a large leap from Oxford around the early 2000s to Stanford, originally for 2 years, and 20 years later, I'm still here. And I think I don't need to tell any of you about the allure of the farm and the excitement of what is possible here. And that really has been the magnet that has kept me here on campus all this time. And I've had just an unbelievable opportunity afforded to me by this wonderful institution. I've had a chance to visit from Stanford, number 10 Downing Street in the UK just last year. I worked with the White House, as you mentioned, and during the Obama administration. I've had a chance to meet many of the technical tech company CEOs here with Bill Gates and Larry Page and Jeff Bezos, all through Stanford connections. Just an unbelievable opportunity afforded to me. I have 3 American children now who make fun of my Scottish accent. This is my little kid. I did promise to answer the question, what was he wearing? And this case, my little boy is also wearing a kilt, obviously traditional dress from Scotland. My last name is Ashley, but in fact, my clan is Fraser. And so Castle Fraser is in the north of Scotland, and several generations of my family go back to Fraser. These days, when I say that, and I don't know if any of you watch TV very much, but there's a TV show called Outlander now that features a character from Clan Fraser, in fact, the main character. So suddenly there's a lot more interest in Scottish history. And this is me in the middle of the pandemic. You see my mask there just a couple of months ago, sitting outside on call for the coronary care unit at Stanford, where I spend my clinical time. On the research side, I'm interested in human genomes and data science. And so I want to tell you for the main time that we have together here about a secret code. It's a secret code that's inside every one of you. It connects you to every living organism on Earth. It connects you to decades, hundreds, even thousands and millions of years of human evolution. And it contains information on your likely skills and talents, your height and weight, your hair color, your eye color. It contains a lot of information about the sort of life that you might live. It is, of course, your human genome. 6 billion base pairs of data is a significant amount more than we're used to dealing with in medicine. 3 billion base pairs, 6 billion data points, 23 pairs of chromosomes. And in fact, it's so much data that if I was to take all the DNA out of every cell in your body, and sorry, a lot of, I don't have control of the screen. It seems to be having a mind of its own here. But if I was to take the DNA out of every cell in your body and lay it end to end, it would essentially stretch to the moon and back. And to put that amount of data in perspective, if you were to stare at this screen every second of every day, 24 hours a day, 7 days a week, it would take you 3 months before you saw the entirety of the human genome pass before your eyes. So this is more data than we're used to seeing in medicine. And that's part of the journey that I want to take you on over the next 20 or 30 minutes. But to get there, we need to go back a little bit in time. And first of all, we're going to go back to the year 2000, when the genetic code of human life was cracked by scientists, which is to say that the draft of the human genome, the first draft, we're now actually, all this time later, approaching a more final draft. The first draft was announced by Francis Collins here, shown in his younger days now, and for many years, of course, the NIH director, and Craig Venter, the rather renegade entrepreneur who challenged the Human Genome public program to a race to see who could finish first. They finished together, and Bill Clinton, president at the time, declared this a draw. To put that in perspective, though, the Human Genome program was funded for $3 billion, and they spent most of it. The Craig Venter genome was about $100 million. And over the course of the next many years, the cost of genome sequencing dropped really dramatically in price. In fact, it dropped so dramatically that it tends to be compared, of course, in Silicon Valley to Moore's Law. And Gordon Moore, of course, with Bob Noyce, the co-founders of Intel. And Gordon Moore himself coined this idea that technology moves so quickly that the number of the transistors on an integrated circuit would double every approximately 18 months. And this became essentially emblematic of the rapid pace of technological advance. So to see the cost of genome sequencing drop at that rate was quite remarkable. But even more remarkable than that, in 2008, what happened was the cost of genome sequencing dropped so quickly that it left Moore's Law in the dirt, to the point that today you can sequence a human genome for just a few $100s. A remarkable and unprecedented change. We're used to technology moving quickly, but this was just really on a different level again. And I show this graph quite a lot. But I also sometimes use a different analogy, which is to say this is a Ferrari that I used to drive past on El Camino Real, just outside Stanford campus. As some of you might remember, there's a Ferrari and Maserati garage just there in Atherton. Not as a professor at Stanford, being in a position to own such a car, I used to wistfully look at it on my way past in my Honda CR-V. And its price tag was just a little beyond the average salary of the Stanford academic, and certainly beyond anything that I could afford. But to put this in perspective, if this Ferrari had dropped in price as much as human genome sequencing had dropped in price, it would very much be in range. In fact, if I slapped down $1, I would get 90 cents of change if this Ferrari had dropped in price at the same rate that genome sequencing had dropped in price. Just an unprecedented change. But the question is, what do we do with that data? And I was having those kinds of thoughts, noticing that genome sequencing was dropping so quickly in price, when I wandered into my future friend's office. This is Steve Quake, who was at the time chair of our bioengineering department at Stanford. And I did not know him well, only by reputation. He was a famous inventor and physicist who had invented a number of things, including a method of sequencing the genome. And in fact, he had applied it to himself and made headlines. This is 2008, that year, when he sequenced his own genome. He sequenced it in his lab in 5 days for about $40,000. And that sounds like a lot. Now I tell you, you can do it for a few hundred, but the genome before his took a team of more than 30 individuals, several months, and cost $200,000. So this was a major advance. And I went to find him in his office in the Clark building on campus, found him on the second floor. It has a very curiously complicated numbering system. I got into his office, which was piled high with magazines and journals, and he was sitting pecking at the keyboard. And we were talking about a seminar we were going to run together, but there was something much more interesting happening, which was on his screen was scrolling by lots of letters. I recognized them as genes and gene variants, and he pulled me over to his screen to take a look. And I asked him what it was, and he said, well, that's my genome. And this was jaw-dropping, because at the time, as I mentioned, I'm a physician at Stanford Cardiology Clinic. We run a center for inherited cardiovascular disease. And at the time, we would send panels of genes. We would get 3 to 8 genes back. It would cost $5,000, and it would take 3 months. And here was somebody not 400 feet away from the clinic, maybe a little further just to walk to Clark building, but showing me his whole genome on the page. And he showed me one particular gene that I recognized as a cause of a heart condition and said, oh, what do you think of that? I've got some variants in that gene. And I understood that myosin binding protein C is a gene that is one of the most common genes that causes hypertrophic cardiomyopathy. So I said, well, I can't tell just by looking at it, but have you got any family history of heart disease? And like most people, he initially said no. And of course, after he thought a little bit and we tried a little harder, we eventually found out he had quite a significant history of heart disease. And in particular, anywhere you see orange on this family tree is a place where Steve's family has some degree of cardiovascular disease or atherosclerotic heart disease. Knowing that he had this variant in a gene for hypertrophic cardiomyopathy, I also asked him, of course, has anyone ever died suddenly? And he told me, well, it's funny you should say that, but his cousin's son, shown here at the bottom of the screen, was 19 years old and never had a sick day really in his life and just did not wake up one morning. And this was a revelatory moment because it was clear that Steve, who I had barely known as I walked in, but otherwise as a famous scientist, was about to become my patient because he needed to come and be seen. And at that moment, we realized that as he was going to come to the cardiology clinic to be screened for this family history of heart disease and this family history of sudden cardiac death, that he was about to become the first patient ever to walk into a clinic with his genome, essentially on a hard drive. And much more terrifyingly, that clinic was actually mine. And we had absolutely no idea what to do with that level of information. As I mentioned at the beginning, we're used to looking at cholesterol tests that have 4 or 5 numbers on them. But here was a genome that has 6 billion base pairs. So how do we begin to think of how we incorporate that level of data into medicine in a way that you could even compress into a 12-minute conversation in primary care? And over the course of the next year, we put together a team of about 30 individuals to try to build the first tools for understanding the genome. And we published them. We included some visualizations and some tables. And fortunately, we managed to rule out that he himself had any significant heart disease. Thankfully, that variant that we saw that first day turned out to be a false positive, which was the best news. We did find some significant things. His lipoprotein little a, that very atherosclerotic, atherosensitive particle, was high, very high for him, and normal being up to about 30, and his number being 156. And so we found a number of things, both in his blood and his genome, to help both him understand why he was at such high risk for heart disease, but also what drugs we might use. And in particular, there are variants that we can use to help inform whether someone is likely to respond to a statin. There's also variants that tell us whether he is likely to have the common side effect that happens with myalgia when you take a statin. In fact, his genome was able to inform that decision, both in telling us what his risk would be, his lifetime risk, not just the risk at that moment in time from a single cholesterol panel, but also the fact that he would likely respond to medication for it, and that he would be less likely to have the side effects. And that medical decision, based on an analysis of an entire genome, was really a first in many ways, certainly for us, and as it turned out, for many others too. And so when we published the paper, we were very happy that to have a fair amount of attention. And NPR did an interview actually with both Steve and I. And this was the beginning of what became for us a 10-year odyssey, essentially, of working on how to bring the genome to medicine, to improve how we do medicine. And we realized people are pretty fascinated by genomes, and we spent the next several weeks answering calls and doing interviews for a variety of stations. And in fact, we were engaged, as I mentioned, by the Obama administration, but later I'll come to that. But at the time, in fact, there was a genome exhibit at the Smithsonian Museum, and they, in fact, featured our work there. So we were very privileged and felt very humbled and lucky. But over the course of the next few months and even years, people would randomly text us photos that they'd seen this familiar face in the exhibit at the Smithsonian as they walked around. Well, one of the people who had seen that work and seen that data was a person called John West. And to tell you about him and his family, I actually have to take you to Buffalo, New York. And I put this on the screen because this, which is to say chicken wings, was the only thing I knew about Buffalo in New York before this call. I'd never been to Buffalo in New York. I'm sure it's a great place, but I certainly knew that chicken wings came from here. And if you're interested in that story, I don't have time to tell you it now, but I think in the endnotes to the book, you can read the story, which is quite fascinating, of how the origin of Buffalo chicken wings was from Buffalo, New York. Buffalo is a pretty interesting place for another couple of reasons. One of them is that the word buffalo, and I didn't know this either prior to this, could be used as both a noun and a verb, which is to say, because buffalo means to intimidate. So you can now make a sentence, in fact, that could get longer and longer with nothing but the word buffalo. So buffalo means buffalo intimidate, and who do they intimidate? Buffalo, 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 if they're intimidating their neighbor. But you don't need to stop there, because if the buffalo come from buffalo, then you have buffalo, 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 and you can go on, and believe me, some people do. So that's an interesting thing about buffalo. But the most interesting thing from the perspective of the human genome has nothing to do with chicken wings or grammatical acrobatics. It has to do with the fact that the origin of the Human Genome Project was Buffalo, New York. And in fact, there was an ad placed in Buffalo for volunteers, and it was answered by 40 individuals. And those individuals came and they donated their DNA for the Human Reference Program. And it turned out that in the end, only 2 people, it was supposed to be 10 randomly chosen, but actually all the DNA for the Human Genome Project came from just 2 people. They're anonymous, although one of them revealed in the book who he was. And he came from Holland. And that's relevant because a few years ago, and you may be familiar with this, a variant in the genome that predisposed to blood clotting was called and named for the city in Holland where it was discovered, Factor V Leiden. And it turned out that this Dutch scientist who donated his DNA in Buffalo and New York to the Human Genome Program had the variant Factor V Leiden. And the Human Genome Reference, which is only not 2 copies of each gene, but just one, so it's one long line of letters, 3 billion letters, actually contained that risk variant, Factor V Leiden. So why am I telling you this? Well, John West, who I mentioned, called me up after seeing our first story on Steve Quake, was an entrepreneur, an innovator, and he'd suffered a pulmonary embolism. And then he'd taken warfarin, as he was told to by his doctor, rigorously every day. And then he had another pulmonary embolism. And he was a bit concerned about this. And so he was very attuned to his own physiology. he was also in a unique position to sequence his genome because he had sold the company Solexa to the company Illumina, now the behemoth at the center of all human genome sequencing that was responsible for that big drop in price. He was the CEO of Solexa, and then he ran the sequencing business for Illumina. So when they started offering sequencing to individuals, the first 10 included 4 members of his family. In fact, he, his wife, and their 2 kids. And his 17-year-old daughter was busy analyzing the genomes of the entire family for her AP Science project, which is very Silicon Valley, of course. And he had called me to say, well, we saw what you did with Steve Quake's genome. Is there any chance you could do the same for ours? And when I said ours, what do you mean? He said, well, we sequenced my entire family, and my daughter and I have been working on some analysis, but we think you can do better. And so, yes, welcome to Silicon Valley. And so we put together the Stanford faculty. Actually, we were working with a group at Harvard as well. And we started to work on a whole family, which was exciting because we only had 1 before. Now we have 4 related people. And so we built tools, and I won't take you through the details, but they're shown here on the screen just to give you a sense of what they look like, that enable us to map the individual DNA fragments from one person to another within a nuclear family, and then to overlay on top of that the genetic variants likely to predispose to disease. And when we were doing that, we realized that within the human genome, there was the factor V Leiden variant. And since we compare the reference with the genome we're looking at and only look at differences, that's pretty critical, because at the beginning, we went to look at his factor V Leiden, the only part of his genome that we already knew, and it didn't show. And so we had to invent essentially a new approach to the reference sequence, and that's what's shown here at the top, the ancestry specific reference. We also built a few other tools, but the main thing was we really cemented the idea that you really need to do genomics in families, because families can tell you so much, and they're so powerful. And that takes us to one really the main topic of, and this is, sorry, just to show it's just coming up slowly. This is features in the Wall Street Journal and Technology Review when we sequenced the family, and John and Anne there, who were doing the analysis for her AP Science project. We also founded a company called Personalis, which is now public and doing extremely well, and has probably the company that has sequenced more genomes at this point than any other private organization in the US. So moving forward well, and John actually became the CEO of that company. So classic Stanford story. But in terms of solving mysteries, which is what's really at the center of the stories in the book, I wanted to tell you about this little girl who came to our attention first when she was 36 weeks of gestation. So she was not yet born. And her heart rate had been measured at 70 beats per minute, which is, okay for probably any of us sitting listening to this talk. But if you're a little baby swimming around in your mother's womb, then your number should be about double that. And so she was transferred to Stanford Children's Hospital with the thought that maybe she was suffering from heart block. As it turned out, when we delivered her by cesarean, we realized actually she was suffering from a particular form and very severe form of long QT syndrome. So the genetic condition where the QT interval on ECG is prolonged by variants and mutations in the potassium-sodium channels. And on her first day of life, this little girl suffered 5 cardiac arrests and was in the neonatal intensive care unit, received CPR each time, fortunately managed to survive. She became, this is torsade de pointes that you may recognize as the ventricular fibrillation rhythm. And here is her X-ray, and showing at the bottom left, as you can see, is a defibrillator. She became the youngest baby ever to have a defibrillator implanted at that time to try and save her. But it wasn't enough to stop the ventricular fibrillation. Because this is a genetic condition, if we could sequence her genome, we could potentially find the cause and give her very precise therapy, whether it's a sodium or a potassium channel version of long QT. Unfortunately, at the time, it still took 3 months and cost thousands of dollars, and essentially, she didn't have that kind of time. So we spoke to Illumina, who'd been working on a faster version of this, and we actually speeded up our computational pipeline. And over the course of a weekend, we managed to sequence her genome, and this is the UPS packet of her genome coming back just 3 days later. We were able to find that this was a potassium channel version of long QT and immediately wean the sodium channel blocker that she'd been given because we just didn't know, and start to make her precision therapy much more precise. We actually found, because we had the whole genome, another variant that had subsequently been shown definitively to cause this early onset of KCNH2, or the hERG channel long QT syndrome, and that's shown here on this on this chart. Happy to say, thankfully, she was discharged from hospital. This is her a few months later. She has a defibrillator still in. She's now 5 years old. I have a newer photo not in this presentation, but she's doing really well. And thankfully, I think is still fairly unaware that she was the baby to have the fastest genome sequence ever performed at that time. Well, here's a stack of notes, and practitioners in the audience, I think, will recognize that most of the time, and this includes for my practice, has been with paper notes, and that many patients, of course, have copies of their notes and carry them around with them, and that's particularly the case if you have a mystery condition that no one can solve. In fact, if you're one of those patients or one of those families, you will accumulate a stack of papers, maybe this high. You'll also accumulate a lot of financial strain and stress and emotional stress and strain as you go from doctor to doctor to doctor trying to find answers for your condition. And there's a reason that this is called the medical Odyssey. It's after, of course, the famous work by Homer, the Odyssey that tells of his 10 to 20 year trek to get back to his homeland in order to gain his wife's trust back from the suitors who were taking up residence in the house. He fought off one-eyed giants and many shipwrecks and serpents and strange animals in order to make it back. And so it's not surprising that given that adventurous journey that the word Odyssey entered our lexicon as meaning that, a long and adventurous journey, and that a medical Odyssey has come to be used in the same kind of way. I like this picture particularly because you might not be able to see this very clearly, but it says the Odyssey, one of the 10 best books of 1996, which I thought was good. Probably it was written just a little beyond that. And if you go on Amazon, I think this is the bit I like the most. You can actually follow the author. I suspect Homer himself might not be, updating with too many new books, but perhaps there are new editions coming together. So medical odysseys. Well, let me tell you one of them. And this story is told in the book, but one of the most stunning medical odysseys that we've had the privilege to be involved in. This is Carson and Chase. Carson and Chase Miller. They are the sons of Nikki and Danny Miller, patients who we came to meet just a couple of years ago. And they both had developed normally at the beginning of their lives. Through their first year, they hit all their milestones. And Carson, the older boy, just as he became a little bit more than one, started to slip back. Where he'd been cruising, he started going back to crawling. Where he'd been able to feed himself, he went back to being needed to be fed. And at that time, Danny and Nikki started to worry that there was something wrong. And they went, first of all, to their Kaiser doctor, and then to a neurologist and a developmental specialist, and then another neurologist. And they were getting nowhere, and nobody could tell them what was wrong. They were given the label cerebral palsy, but were told just to keep watching. There was nothing that could be done. And when Chase, Carson's younger brother, was born, and he started having some of the same developmental challenges, it became very clear this must be a genetic disease. And they even had their exome sequenced, which is the 2% of a genome that represents the genes, and no answer was found. And they had by this time accumulated years of nights up late at night, searching the internet, desperate for answers, looking through the medical histories, looking through the medical tests, emailing everyone they could find. Danny spent literally hours every night trying to do this, trying to find an answer. But no answer could be found, and they'd seen 5 doctors, including 3 neurologists at the time, when they first heard of the Undiagnosed Diseases Network. And this is a network we've been privileged to be part of and even lead at Stanford. And it's where we take on the hardest cases in medicine. And our principal tool is sequencing the genome. And so Carson and Chase came to our clinic at Stanford, and we sequenced the whole family, just like I mentioned for the West family, the 2 parents and the 2 kids. Instead of just the exome, the 2% of the genome that is represented by genes, we sequenced the whole thing. And the power of those 2 things coming together allowed us to make a diagnosis, which is to say that on these sheets, and these are the actual genome data that you can see for mother, father, and the 2 siblings, we see that a variant in the same gene that came from mom, and over on this side, the same gene, a different variant that came from dad, came together in both of the boys to cause this condition. And this condition was a condition that only 7 other individuals in the world had ever been described with. And it's called MEPAN syndrome, and it's caused by a variant, in this case, 2 variants, in this gene MECR, M-E-C-R. And that's part of this relatively unknown or little known mitochondrial metabolic pathway. Shown here, I won't be testing anyone in the audience on it. I doubt I could draw it myself. On the other hand, just to remember then, that if you take out the MECR enzyme shown here, then you disrupt this metabolic pathway at this point, and you start to have a deficiency of the downstream components. And in particular, you have a deficiency of lipoic acid, a really important intermediate metabolite. And so Danny now has the information. They have the information about what causes the disease. Their diagnostic odyssey is over thanks to the genome. But that, of course, isn't enough. It's a lot for these individuals, but what happens next? How can you use that information in an action to make the lives of these little kids better? Well, Danny was essentially Googling this one night, and he said, well, if lipoic acid's what they're missing because of the absence of this gene, and we don't yet have gene therapy, maybe I can fill that in. So he Googled it. And what came up was the finding that lipoic acid actually exists as a supplement that you can buy on Amazon for $16.10. So the power of the genome here to give him the answer essentially told him where to go looking. And while this isn't like a gene therapy that completely restores function, the boys have now been on this for more than a year and have started to hit their milestones again. So a truly remarkable story of the power of the genome, of working together, of sharing data. And the family were kind enough to be featured in a couple of different news stories. This is them on CBS This Morning, Carson and Chase shown there with their dad, Danny. And there's a focus there on the work we did with the Undiagnosed Diseases Network, and together, that was just one story, but over the course of the first 2 years of the network, we solved 132 cases, that's about 40% of the cases, and identified 31 new medical syndromes. And you're welcome to look up this paper in the New England Journal of Medicine, or many of the other now hundreds of papers from the network, and we continue to do this work. Kind of Sherlock Holmes sort of approach. Sometimes it's all about the clinical work, sometimes we don't even need the genome. 3/4 of the time we use the genome to make the diagnosis. Let's see. All right, well, just for the last few minutes then, I wanted to tell you about a different genome. I think it's just coming up on the screen. Oop, I think we've skipped past it. So I've ruined the surprise. But let's see, I didn't quite catch, maybe we'll see if we can go back. But on the screen, you will see in a moment, a different genome. And this is, in fact, the proteins that map to the genome. And the genome in this case is not 6 billion base pairs of DNA, but rather 30,000 base pairs of RNA. And this is the genome. I think I've lost control. This is the genome of SARS-CoV-2. We can't hardly give a talk in this year in the pandemic without talking about what Peter Medawar described as a piece of bad news wrapped up in protein. And I don't think we'd have too many people arguing with that characterization of a virus in 2020 or 2021. But I want to tell you here just the extent to which, despite the disastrous response of many governments around the world and the real inadequacy of the political response in many places to the pandemic, resulting in way too many deaths, that there is a silver lining. And part of that silver lining comes from the triumph of science. Because if you read back into 1918 with the flu pandemic, that was a horrendous situation where 50, maybe even as many as 100 million people died. But if you look and read about it, what you hear is all the same things. There's questions about masks, about hand washing, there's skepticism, there's people who don't, there's conspiracy theories about what helps aspirin, what doesn't help back in the day. All of that is so familiar. Humans have changed so little. But what has changed in 100 years is science. And whereas then we were in the dark, we barely knew what there was to deal with it. This year, because of genomics, we both know what the virus is. We can spell out its genome. And within hours of doing that, January the 10th last year, literally hours, there were clinical tests derived that were aimed at and developing, aimed at diagnosing the condition on an individual basis. And many of you may have had one of these rather painful nasal swab tests. The individual testing that you can see, this is Ben Pinsky from Stanford's virology lab, and Stanford was one of the earliest to have an individual clinical test for the condition. But we can also use sequencing in order to do early warning detection systems. And these are systems that we should have been in place before this, but really weren't. Here you can see on the graph in the blue and green, the amount of SARS-CoV-2 genome that was found in the wastewater for a treatment plant in San Jose. And you can see that by 1 week, even up to 2 weeks, preempts the increase in hospital admissions. So we have an early warning system detection that we can use sequencing for. We have individual diagnostics that we can use sequencing for. And one of the places where, unfortunately, due to regulatory challenges, where things have been a little bit harder, has been in home testing. But we're working with Manu Prakash, a MacArthur Award-winning bioengineer at Stanford, to develop and push forward with this $1 30-minute at-home test based on saliva. And we've been piloting this in Stanford over the last month or so, and are now looking to spin this out into a company to bring it rapidly at the millions and even tens of millions or hundreds of millions of tests much more broadly. Of course, the really important thing about sequencing the genome of the virus was the age of genomic vaccines, and whereas there are traditional vaccines coming, almost all of the ones that you will have heard about from BioNTech and Pfizer, from the NIH and Moderna, from the University of Oxford and AstraZeneca, or from Johnson & Johnson, all of these are new age genomic vaccines based either on mRNA or DNA with a viral vector. This is the age, essentially, of genomic vaccines, and it is spectacularly effective, not just in speed, and this is just unprecedented. You'll remember that some people were saying 9 years was the fastest a vaccine had ever been developed before. Other people had said 4 years. Well, the virus was first sequenced on January the 10th. By January the 15th, an mRNA vaccine was designed. The phase 1 and 2 trials began less than 2 months later. And basically about 6 or 7 months after that, they were complete, some of the largest vaccine trials ever performed. So if there's a silver lining to this pandemic, it is the spectacular speed and unbelievable efficacy of these vaccines. Not only is each one of these primary candidates 100% effective at reducing death and hospitalization, something that we focus less on, but that's the real endpoint. Every single one of those, 100% efficacious. But some of them were even 95% efficacious at preventing any symptomatic disease at all. So that's eradicating even mild cold-like symptoms. The other two, the J&J and AstraZeneca, are still in the 70 to 80% range for reducing even down to mild cold symptoms. So just an unbelievable speed and efficacy for these vaccines. How else can we use the human genome? Well, we've been using it at Stanford to look and reveal disparities within the populations of those who came to Stanford. This was the first 1,500 patients who tested positive and their genome by genome segment ancestry. And it revealed what many of us knew, that those who are most underrepresented in society were those most overrepresented in the patients who tested positive for COVID, particularly Pacific Islanders and Native Americans and those with Hispanic heritage. And that points us to where we need to attend to make sure that equity and disparity are really the focus of our efforts in ending this pandemic. My last message then, I'd love to hear your questions, but the science has given us all these tools, and genomics specifically gave us the ability to find the virus, to sequence the virus, to track the virus, to develop vaccines against the virus in record time. It's just a remarkable change, and the main change, as I say, that you would notice from reading about the 1918 pandemic. Well, it's been my absolute pleasure and privilege to share a few stories with you today over lunch or whatever time zone that you're in. I look forward to a few questions, and I hope that you enjoyed this. Thank you.
Terrific. Thank you, Dr. Actually, take a breath here as we start looking at some of the questions that people have in the last few minutes that we have. I think one of the things that people, felt that back in earlier in the in the 2000s, when the genome was finally mapped out, and they said, we've discovered this all, but it is an endless journey to keep identifying another piece and another piece. Is that correct? It wasn't finished at that point. It was just the general construct was defined, but is it an endless task to continue the mapping?
Yeah, it's not quite, but sometimes the analogy we use for that is like painting when I was in Scotland, it was the Forth Bridge, which is the bridge over the river in Edinburgh. And here, I'm sure it's the Golden Gate Bridge. But the idea that, you only just finish painting one side and you have to go start at the beginning instead of endless effort. I'd say that's not quite right, but for sure, over the last 20 years, that does describe where we've been, because that sequence in 2001 was really just the draft sequence, and there were a lot of holes in it. at the time. I mean, it was remarkable, and it had essentially most of the genes and the rest of the genome fairly well covered. But what has happened, and it stayed the same for a few years later, maybe there was a sequence that was like a second or third draft. And then nothing much changed for the last 10 years. We got a little better at understanding variation in multiple humans, so beyond just the one reference. But with newer technology that I didn't have too much time to talk about today, long read sequencing, we've started to fill in the holes, and particularly the ends of the chromosomes, which were hard to cover, and areas that we call low complexity. They're actually high complexity because they're hard to sequence properly. And actually over the course of the last just 2 years, there's been a rapid acceleration in our ability to close those gaps, such that we really are looking at what you would essentially call a finished genome any day. And I think after that, we'll move on to more like a population map of genomes. I think that's where we'll go next. And we will finally, I think, lay the Human Genome Project to the side a little bit as we start to map whole populations of people. But the questioner is exactly right in saying that the draft that was produced really was an early draft, and we've been working on it pretty much constantly for the last 20 years.
Terrific. So some of the examples that you shared with us were very interesting in terms of finding obscure problems and then being able to immediately change the plan for treatment or the therapeutics that were used with these items. So, we're all jealous about that. Yeah. When's the point that your physician is going to come in and at the very beginning, as you have problems, do a genetic analysis and really personalize your medical treatment?
Yeah, now that's important. So obviously I've given examples from rare disease. I think one of the first things to remember is that rare disease is individually rare, but collectively very common. Actually, it's remarkable. Every time I say this statistic out loud, I almost don't believe it, but 1 in 15 people actually has a rare disease, which means that collectively, rare disease is as common as diabetes. But of course, that still leaves 14 out of 15 people for whom the genome right now is not touching them. So 2 things, first of all, for the rare disease, I do think we'll move to the point where it's available much earlier. And the moment that there's a problem, you could have your genome sequenced. There was a program called BabySeq that had a publication last year where the idea was maybe people wanted to sequence their whole genome at birth. We actually have another company that was mentioned at the beginning called DeepCell that can look at fetal cells, isolate them from the maternal circulation and potentially sequence them even before birth. I personally don't think that many people will want that, but it's certainly technologically possible now. I think maybe though what the question was getting at was this: what about the other 14 of the 15 people without the rare disease? What about people who are at risk for cancer and cardiovascular disease like heart attacks and high blood pressure? When is the genome relevant for them? And again, I think although it didn't happen as fast as we thought, that Steve Quake's genome was 2009, 2010, and we thought within 2 or 3 years, everybody would have their genome sequenced. But in reality, I think things always take a bit longer than you imagine. And while we made these great strides in rare disease, it's really only, to be honest, this year. And in fact, just last week, we rolled out at Stanford the whole genome in the cardiovascular clinic in a way that includes something called a polygenic risk score, which is to say that we can now add up, in fact, millions of variants across a whole genome to improve your risk prediction for heart attack and for cancer. And so really starting this year, and this is at Stanford, as probably not too many people on the call would be surprised to say we're kind of ahead, and one of the first health systems to do this are rolling out these polygenic risk scores that will allow us to predict the risk and really incorporate the genetic risk to the standard risk factors. Let's say for heart attack, of course, we're thinking smoking and cholesterol and diabetes and high blood pressure, but we ignore the family history part right now. Even the formal prediction model from the SCVD, from the American College of Cardiology, ignores that. But now not only do we not ignore it any longer, but we put real quantification around it so that you have a genomically enhanced risk score. And that's the basis on which we will be making decisions going forward for things like statins and other preventive therapies. So I think we're finally there. I think it's a really exciting moment.
So as a follow-up to that, as people have that opportunity to, shall we say, assess their risk or know what their risk factors are or know how their genetics influence how they might react to certain medicines. What are the risks about personal privacy, or insurance companies, or people saying, I'm not going to hire you because you might have a heart attack, or other people saying, that suddenly my genome is out there and there's a risk to somehow my privacy? What are your comments
Yeah,
around that area?
yeah. Well, there's two aspects to that. I think the first is, yeah, there are risk factors for diseases, and the genome is among them. And so if we add them in, there's one part of the question is, yeah, is there an issue where people will be discriminated against in the same way that they might be if they drive erratically by an insurance company for driving, or if their lifestyle factors are a certain way, it's possible for health insurance companies to distinguish in that way. We obviously don't approve of that or encourage it in any way, but it's certainly possible. And that's a tricky question, but not one that is new to us. I mean, that's really no different from whether it's a traditional risk factor or the genome. Of course, what is different with the genome, first of all, is the size of the data. I've talked about that, but also the personal identifiability. And that's what I think the other part that the questioner is getting at, in that although it's a very complicated signature, your genome is a signature that is identical and specific to you, as I touched on at the very beginning, in a way that your cholesterol number isn't. If your LDL is 96, then there's lots of people who that doesn't identify you in any way. But if I grab a few thousand base pairs of your genome, that would be enough to identify you. And so we take that concept very, very seriously. And we, in fact, at that very first study, I didn't talk about it, but we actually had a whole ethics group that were involved and published a separate parallel paper at the time we did Steve Quake's genome. And that group have continued to work over the next 10 years to inform all our policy decisions around when to use the data, where the data would be, where it would be stored. And the cloud providers, we predominantly put the data now on the cloud, have been very, very attentive to the security concerns. And remember that these are the same providers who work with banks and who work with the Defense Department. And so we do have that kind of level of security, and we really do pay attention to it. So it's a very important question.
What do you get most, it's sort of a personal question here for you, as what is your real optimism about, and your excitement when you see where you might have been 10 years ago in your career and understanding, genomics and cardiology, or whatever the particular specialty was, and where you are now? How has your perspective changed?
Yeah, it's a great question. And in fact, the final chapter in the book, I sort of address that a little bit. And I start with this quote that I love from Bill Gates. And he was sort of a hero of mine growing up and led me, to move to program those computers when I should have been out playing. And that's really what drew me to Stanford originally. But he has this quote that says, most people overestimate what they can do in 1 year, not surprising, but underestimate what they can do in 10. And I think that given it is now a little over 10 years that we've been working on this, I think that really rings true. We thought in 1 year everything would change, and really things moved quite slowly at the beginning, but they really picked up pace to the point where really, when I look back now and think that we, as I mentioned just in the last few weeks, have launched this genome sequencing service for all patients at Stanford with inherited heart disease. We're bringing preventive genomics through these polygenic scores I mentioned in a few months, and then later in the fall for heart disease and for cancer. I think we've really come a long way. We're working on, and I mentioned the FAST story where we could do genomes in 2 or 3 days. Well, just 2 weeks ago, and don't tell anyone because we haven't really made this public yet, but we actually used a new approach, which includes these long-read sequencing, to sequence a genome in just 3 hours. And so it's just, I think many of us, I'm sure in the audience, were attracted to Stanford because of this unbelievable technological advance. I mean, we marry that with real sense of compassion and understanding the patient and thinking about traditional tools of medicine these days. We really try to do both. And in this undiagnosed diseases program and in genomics generally, that's what really lights my fire. The ability to combine these traditional tools of medicine, which are taking a history, understanding the patient, listening carefully, thinking about the family and how everything around the patient impacts how they present. But the ability to take those very traditional tools of medicine, hundreds, thousands of years old, and combine them with the absolute cutting edge, things that are not available to anyone else in the world. Well, that's Stanford in a nutshell, and I just couldn't be happier to be here and be part of it.
Terrific. Well, I think that is about, I'm sorry that I know that there are a few other questions that we didn't get to, but I do want to thank Dr. Ashley, and this is fascinating work. It's something that each of our new MD students start learning about at the beginning of their careers, where many of you, you know, it was not part of your training back in the day. And that just shows us that medicine and science continues to evolve and work. Anthony, could you show us once again this slide of Dr. Ashley's new book, the Genome Odyssey? Oh, here we go. One moment, please. And the reason that I want to put the book on the screen is not only to acknowledge Dr. Ashley's work and call your attention to the fact that this book is just being released in the next week. However, for all of those of you who are on the call today, Stanford Medicine Alumni Association is sending you a free copy of this book. We have worked with Ewan's publisher, and they have allowed us to get access, and we will be sending them off in the mail in the coming weeks, so you can read more about the story that we have seen today, some of the stories, and then other medical mysteries that he's uncovered during this journey. And it is an odyssey, because there are you know, tragedies and challenges and triumphs throughout, with the typical hero's journey across studying about the genome. Also, I want to call your attention to the fact that there will be a recording of today's webinar available on our website in the coming days. Just give us about a day or so, and then we will post it at med.alumni.stanfordmedicine.com or .edu, I'm sorry. And then please take a moment to fill out our program survey. We like to send surveys after each of our programs, including the Half Century Club, so that everyone can tell us how to make things better for you.